warm welcome to Living Science. It is my great pleasure to introduce Dr. Sudhir Kumar Sopory, our guest today. He was the Vice Chancellor of JNU from 2011 to 2016 and the Interim Director of ICGB New Delhi from 2014 to 2015. Dr. Sopory has made several outstanding contributions in the area of plant biology with his research work on nitrate reductase, glyoxalase and improving plant breeding methodologies. He has published over 200 papers in international journals and continues to be actively involved in research. Dr. Sopory has received numerous awards in recognition of his work, including Padma Shri by Government of India in 2007 and Shanti Swarup Bhatnagar Award in 1987. He is the fellow of many Indian and international science academies. Apart from his academic accomplishments, what strikes most about Dr. Sopory is his humble nature and approachable personality. He believes in open door policy and finds time to discuss academics or any other issue that concerns students. Above all, Dr. Sopory has the unique ability to express most opposing views with extraordinary calmness. We are very happy that Dr. Sopory is with us today. So Dr. Sopory, thank you so much for being with us.
My pleasure.
And you spending your considerable amount of time on Saturday with us, it means a lot. Your career you started from JNU, actually, where you became Vice Chancellor later. So your PhD is from Delhi University with SC Maheshwari. And when you became faculty at JNU, you did a considerable amount of teaching, undergrad and postgrad. So our question at this point is, we want to know how much influence does teaching has on research in terms of maintaining enthusiasm and looking for new questions.
I think I did do undergraduate teaching in JNU. Didn't have an undergraduate teaching. I directly went into postgraduate teaching. But earlier in my career, when I was doing my PhD, started my PhD, and that time there were some issues in the university. I had to leave, and I did undergraduate teaching in Merit College because there were some problems, and it's only after teaching for more than 7-8 months that I returned back to Delhi University to complete my PhD. So there was some experience of that undergraduate teaching and the postgraduate teaching which I did in JNU. I personally have always felt that teaching is not a job, it's not a profession. Teaching has to be passion, and teaching has to be, a teacher has to keep learning. If you go into the class, either it's undergraduate or postgraduate, and you have this impression that I know everything, then you can't be a good teacher. You must realize your limitations in giving a lecture. The second thing which is very important is that especially nowadays, the scenario which is there in the universities and other places, everybody in the classroom doesn't have an equal understanding, IQs, how much he can learn, because they are coming from different backgrounds, different schools, different colleges. So teacher must see that what he is teaching to the highest level of the student, with his, whether the other one is also following that one.
Yeah.
So I think that alertness of a teacher has to be there. And I feel teaching has a great influence on research. It influenced my also, because the questions which the students can ask, in fact you have to encourage. It has to be always, a teaching has to be two-way process. You teach and you ask them to ask questions on that subject. We don't have to feel very embarrassing if you can't answer a question. I used to keep a record of all the questions, which students ask, and then go to the library, look at the answers if they are available. So 1 or 2 classes will be only to answer those questions. And that was the process by which you learn. And when you learn, you can also direct your research, because the kind of questions you ask. That's what I was telling, that questioning is very, very important in this one. What kind of questions you ask. And teaching, of course, has to be of that kind where a teacher can pick up the talent. I don't know what talent means. I will put it in the inverted commas. Talent, because in the United States, I don't know if any one of you have read two recent articles in Nature. One was which published in May, and another only the last week it came up. That how do you nurture talent?
Oh.
And they have done a big studies, especially in Vanderbilt and others, where they are creating these small schools or centers where they pick up people based on a test which is given to them.
Oh.
And then they filter out some of those people, bring them into this center, and how to nurture them further so that they become leaders in their own fields. It's not only for science and technology, but for many other things they are doing it. And these are basically the skill development or the mathematics which they are really talking about. A lot of research which has gone into it. Of course, learning and teaching is you learn in a certain social milieu. Everybody comes from a different social background, economic background at a different level. But there are also this other paper which I don't totally agree with that, where he's talking about the genetic element.
Oh.
And they have said they have taken about more than 2 lakh people to look at if there are certain genetic loci which are associated with educational attainment. So they have mapped 76 loci where they say if these loci are present, this gives to. So they're saying that 20% is genetic according to this. Rest is how you teach, how you bring them up and other related issues. So teaching is a science by itself. How you teach, how you learn, how you bring people up, how you identify the leaders, how you groom them up. So university and colleges have a major responsibility on these issues. So I personally feel, I personally like teaching more than research. But I don't have that opportunity anymore. When I was in JNU, I would rather leave lab, but I can't leave my class. And this happened, when my son was to be born, I just left my wife in the Safdarjung hospital, and doctor said it will take 2-3 days or something. So I came back. I said, you be in hospital, because I have my classes, I can't leave my class. And when I was taking my class, one of my students, Neeraj Datta, unfortunately he is no more, he passed away. He said, sir, son is born. I said, ask them to wait, I'll finish class and go. वो भी एक पागलपन था. But anyway, that is the way it has to be. You can't. So teaching as a teacher has a great influence, and vice versa. Many times you remember students, but you can't remember all the students. But you remember some students in a class of 50 or 100. So that's the other influence which you have. So that day there was a teachers' day celebration in JNU. So he said, you remember good teachers, but I said we also remember bad teachers. So both ways you remember. So teaching is very, very important.
So when you started your career, research career in JNU, so you were working on understanding the process of seeding, and you studied effect of nitrate and light together, and more of nitrate. So can we know that little bit of that work?
After finishing my PhD, where I was working mainly on tissue culture, other related things, and also on postdoc, some work was done. When I joined JNU, I was told that extremely good facilities, sky is the limit. But when I reached there, I thought it was only the sky, the limit was not there, because the labs were almost empty, and there was hardly anything which one could do, except for small pH meters and other things which we started buying. There was a strike by students. I was the member of the academic council. A strike by the students and some faculty that you have brought us here, because that was the first batch of students. So what is expected of us? And I don't know if many of you may not have been born at that time. This was 73. So there was little financial problems with the government of India. There was no dollar exchanges which were possible in those days. So we had to buy all our equipment through East Germany or East European countries. So all those kinds of equipment came much later. We had one student, that's what I say, you learn from students. So we said, under these conditions, what can we do? Because I had some interest in how plants perceive environmental signals like light or other signals, and how light is perceived and it regulates plant development and biochemical responses. So he had, he was a student from Delhi University, but had worked for few months in Tata Institute with Professor Johri, and he had done some work on nitrate reductase. So he told me that, can we work on nitrate reductase? And Narayan Yadav, who later on became one of the chief of DuPont laboratories in US, because he did his PhD abroad. So I said, how do we do nitrate reductase? For enzyme extractions, you have to have cold room, you have to have other things. He said, sir, I have read one paper where you can do in vivo assays of the enzymes. So you don't need cold room. You can do it in ice buckets and other things small. So that's how it started. So we started doing in vivo. Because we need to do something, you can't be sitting idle. So we started working on nitrate reductase. By doing in vivo assays, there was no cold room and others. Now how do we work on the light effect on nitrate reductase enzyme activity? Now to work on light, you have to work in darkness. Now there was no dark room. So how do you do? So what we did, we converted a small room, put some black curtains, but even then the light comes in, there is some peeping of the light, and plants are extremely sensitive to light, and they can disturb your experiment. If you are working in the darkness, you are given a short treatment of light to check what's happening, but in between if there is any other light coming in, that can disturb the experiment. So he said, sir, this is not a good dark room, so what do we do? I said, we'll work in the night. So I used to stay near IIT gate, so I used to walk in the night. So all our experiments used to be during the midnight, and there is no light outside. And we did all these experiments in the early years of 73, 74, 75 on nitrate reductase. So basically what we found that nitrate reductase activity is regulated by light, and light is perceived by photoreceptors. Like we have photoreceptors in our eyes, plants have photoreceptors, and that time only one photoreceptor was known, which is phytochrome. It is only later on they found many photoreceptors, blue light receptors and others. So we looked at whether phytochrome was involved in this. And for this you need different wavelengths of light, that is red light and far red light, because phytochrome is a light switch which gets activated by one set of light and deactivated by others. Now doing these sets of experiments, you need filters, we need the filters. So we got some red papers, red and blue papers, some table lamps, worked through that, and were able to establish initial experiments that it is the phytochrome which perceives the stimulus from the environment in order to regulate nitrate reductase activity. And nitrate reductase is a very interesting enzyme because it is stimulated by its substrate. Nitrate reductase converts nitrate to nitrite, and nitrite reductase converts nitrite to ammonia for further converting into amino acids and all that. So this is the first step of that pathway. And we also found the second step is also regulated by light. So when you give nitrate and light together, you are able to induce this activity. Later on, of course, when we went into cloning of the genes, purification of enzymes, we found that once plant perceive light, and this is perceived by phytochrome, this pigment in turn certain signals, and these signals are either calcium, which we found, or also some lipid signals, phosphoinositide signals. And these signals in turn activate the expression of the genes of nitrate reductase, and that really increases the activity of the enzyme. More protein and more enzyme activity. So whole this pathway from perception to gene expression was what kept us busy for almost 5-6 years or even more. because we looked into all the other related enzymes and pathways. And by the time people had isolated other phytochromes, so the task became even more difficult, whether other pigments are involved and all. But one thing which happens, maybe some of you would know, that when plants perceive light and is perceived by phytochrome, there is one form of phytochrome which gets degraded by itself. So it's a negative regulation. So light negatively regulates phytochrome because it degrades, and positively regulates another gene like nitrate reductase.
Okay.
So one of the students who worked in detail, where does this bifurcation occurs? So you have a nitrate reductase gene, you have phytochrome reductase gene. Signal is same.
Light.
The signal is same, up to certain point it moves same. One of the signal goes and it upregulates, the other signal downregulates. So how this diversification occurs, and that's what we're looking into more details. So we knew that one phosphoinositide cycle or calcium cycles which are regulating these processes. So I think this nitrate reductase story, phytochrome, other related enzymes which we worked on, kept us busy for many years because the technology kept on evolving, cloning and other things came much later. And some of those one of the students is still continuing on this work. He is professor at Indraprastha University now, where he continues with nitrate reductase. But the other thing which has come up lately, that nitrate itself is a signal. So there is a nitrate, these are called the nutrient signaling pathway. So nitrate signaling pathway also comes up. So you have a light signaling coming, you have a nitrate signaling coming. How do they talk to each other? The crosstalk, and then induces the expression of certain genes. So those are the questions which I had in mind, and kept on working on this for many years.
So now we understand light is important and it's someone else's work, but you were talking before, if we like to know a little bit more. So for flowering, darkness is important.
Yeah.
Can you just share that a little bit with us?
Plants depend on light not only for photosynthesis, but it also depends for all its developmental responses. I think without light, nothing can work. There is in our scriptures, they said, how did the life really, plant life arrive? So there were many people who were dying that time. They went to Lord, that kindly save us, what are we going to do? And he requested sun god, give sun, and through your child, which is moon, give water. So sun and moon, one gives light, another gives water, and the soil, which is the Prithvi, which they said, will give all the nutrition, and then all the seeds started germinating and people were saved. So this is written in our scriptures long back. So it's the sunlight, water, and Mother Earth which creates life on this universe. So light has many different problems, not only in terms of its development of plants, but as you rightly said, in flowering. So both, there are different kinds of plants which are short day plants, long day plants. They require different time of light period and darkness to do it. But it was found many years back, I think in 30s and 40s, that it is the darkness which is more important for flowering than the light period. Sleep is important. You have to sleep properly. And if you disturb the sleep of the plant, not that they are really sleeping, even in darkness they are doing a lot of their work. A lot of work goes on there. Not like us that we totally relax during night period. So they keep performing their biochemical functions, physiological functions. Somewhere that dark period becomes extremely important for flowering. And the experiments where if you disturb that dark period, even for a shorter duration of time, then the plants will not flower. And the duration of disturbance can be even to the tune of seconds in certain plants. If you disturb that dark period even by 5 seconds, then the plants don't flower. So that's where the plants are extremely sensitive to external environmental conditions. Preparedness.
Very nice.
That's what I call them, awareness in plants. People think awareness is related to consciousness, but I feel I don't call plants have consciousness, but I say plants have awareness of some form or the other. They know what's happening outside.
So they have awareness. Do they have intelligence? So is there enough data now in plant field that we can say the plants have memory or inherent intelligence? So what is your opinion on that? Because some people would argue plants
are intelligent, but whether they have intelligence, I don't know. I would put it that way. But yes, memory is something which is coming up in literature in a big way. That plants probably have, in some form or the other, they store some memory. In fact, some of the experiments which we had done long back was on, this was the same light and nitrate reductase experiments, because both of these are required for induction of the gene of nitrate reductase. You need light, you also need nitrate. So what we had done, we separated these two inducers. You give light first for 5 minutes, and after a gap of some time of darkness, you give nitrate, and then it induces. But after giving a period of light, if you increase the darkness beyond 8 hours, then they don't induce. That means up to 8 hours they were able to retain some memory that they had seen a short period of light, which is lost. Now what is this kind of a memory? We don't know. But what people are working now, for example, a lot of papers coming what is called the stress memory. When a plant has seen a certain level of stress beginning, maybe it has seen for 24 hours or 12 hours, they prepare themselves. After that there is no stress. But if the stress comes later in the life cycle of the plant, they remember, and such plants behave much better than those who have seen the stress for the first time. So this kind of a stress memory or light memory does exist in the plant. Now what is the form of this memory is something which people are trying to work out, whether these are not genetic changes, but mostly probably some epigenetic changes which are there, or these could be some kind of a proteins which are present, prions which are there in animal systems, which are also related to memory mechanisms, because they can form certain complexes. And this year there was a paper in PNAS which had, for the first paper we say prions do exist in plants also. So those memory molecules which are present in animals are present. Of course, our own work, we had shown that a lot of neurotransmitters are present in the plants, like serotonin, 5-hydroxytryptamine. Earlier people had shown acetylcholine. These are all present in the plants. And our experiments of like these where we had separated light and nitrate. Now if you don't give plants light at all, but what we gave is serotonin, which is a neurotransmitter. So in total absence of light, you give the induction by a neurotransmitter and then give nitrate, it gets induced. So whether light induces these kinds of transmitters, which are the storage, memory storage, we don't know.
Right.
I mean, this is the kind of research. We also found electrical signaling in plants, which move from one organ to others. But all this work came to a stop when I moved to ICGB. I want to restart sometimes. That's why the difference between universities and institutions, that in institutions you get work in
narrowed
a specific
and focused.
But in university nobody would ask me questions, whether I ask a useless question or a good question. Just for fulfilling our curiosity, we kept on asking few questions and working on that.
And last time when we met, you also talked about memory in context of mimosa plant, and they actually react.
Mimosa plant's memory, yes. Recently there's a group in Australia which has been working on this. All of you know mimosa plant? I don't have a picture here, but if you touch that plant, its leaves collapse. Now why do its leaves collapse? Oh, that is a mechanism. But what advantage it is to the plants, I mean, many people even don't think about it, why the leaves collapse, in many of the mimosa, which have a very, very fast leaf movements. It's because underneath the leaves, there are spines, small spines. So when you touch it, it gets that fear that you are going to kill it. So what it does, it puts the leaves down and puts the spines little up so that it pricks you will take your hand back. It's a defense mechanism. Very, very interesting defense mechanism. This is in many other plants also you have similar kind of a situation. Now in this what experiment was done, this experiment in Australia, she grew this mimosa plant in a pot and taken a stand, and in the stand there is a place where you put the pot. So what she does, she throws this pot from top to the bottom. Pot doesn't break because there is some cushion there. So leaves collapse. Leaves collapse, but they know that nobody has touched. There is no danger. So he does it second time. She puts it third time. They collapse. But when fourth time she puts it down, and they don't collapse because they have no fear. They think it's somebody is cheating me. Nobody is going to kill me. So after that they don't collapse. Now this memory, they keep it. That I was dropped 3-4 times, nothing happened, and now why should I close my leaves? And later, it's not that they have lost that capacity of bending those leaves, because if you now touch it with finger, they will collapse.
They can differentiate between the two.
So they feel there is no problem with us, so I don't need to collapse. So these are kind of experiments which many people are doing, whether it's a light memory, stress related memories, in memory related to flowering, which people have seen. I don't know how many of you have seen apple plant, apple tree. You have seen. I used to live in Srinagar, so I have seen it. Apples grow there. Normally, we say that the stimulus for flowering moves from leaf to the apical meristem, and where the flowering meristem is formed. So the vegetative meristem gets converted to flowering meristem when a stimulus goes from leaf to the apical meristem. Now that means leaves are required for flowering. And what is this signal? Many people have worked on it, and now they think it's probably RNA, which is a signal, not a protein or any other chemical compound. Now in many of these plants, what they find that there are no leaves. When apple tree starts flowering, there are no leaves. There are buds from which the flower starts. Leaves come much later. That means the previous season, when the winter season was there is some kind of a stimulus which was already produced, which has been stored in some kind of a memory, and that is used when the spring comes and they start flowering. So these are all unrelated experiments, many different ones, giving some idea that there may be inverted commas memory, not our kind of memory, some memory, some chemicals, some proteins, microRNAs, epigenetic changes which may be there, which can be called as plant memory mechanisms. Interesting area to look at, but you need the right kind of system to work on.
Very interesting. So you talked about, you looked into effect of serotonin, and also another animal pathway which also exists in plants is glyoxalase pathway, which you have studied extensively and contributed significantly in our understanding of what this pathway does in plant. It's a abiotic stress managing pathway. Can you discuss that work with us? We'd love to know. It was known for 100 years when you actually started working on it. So what made you think, you can start on, what excited you to think on?
I was working in Glyoxalase even in JNU, but from a different perspective. And this is again when I got a feedback from the student. My whole career, is that's you are the people who give ideas. There was a student who was working in cancer laboratory. There was Professor Rao in JNU was working on cancers. So the student was working on cancer, and they had got some idea that glyoxalase enzyme somewhere is related to cancers.
Okay.
And he gave me a book which is called Electronic Theory of Cancer. And this was written by Albert St. George, who was a Nobel laureate. Now his idea was that cancer cells basically means proliferating cells, and those which are non-cancer cells are differentiating cells. Okay. Now, my experience in Delhi University when I used to work on tissue culture, in tissue culture, if some of your plant scientists here, they would know that you put any explant into the test tube and it starts proliferating and making callus. Now, I thought callus is a cancer and the non-differentiated part is a non-cancer. So, his hypothesis of methyl glycol and glyoxalase. Methyl glycol is a toxic compound which can block cell division, and glyoxalase is the enzyme which detoxify methyl glycol. So, it can remove toxicity. So, it is a question of balance between toxic compound and the enzyme which brings the toxic down. So, I thought can we try it in callus system, whether the similar thing is happening. So, there were students who started working on this, and we found some correlations. At that time we did not have protein purification or cloning which was going on, and so he showed that some kind of a correlation does exist. This was mainly indirect evidences using some of the inhibitors of glyoxalases and other things. इसमें जैसे my supervisor. So I came from Kashmir, वहाँ पे इतनी पढ़ाई वढ़ाई इतना उतना नहीं था, ठीक था, but I didn't know many things. So I went to him after being rejected in many places. So I finally landed in his lab, because I didn't want to get into all this PhD. So he said, I can accept you to be my student. You will have to work in the lab without fellowship for 3 to 4 months and prove to me that you understand what we are doing here. Enthusiasm karunga. So after he gave me a topic, because they were doing RNA extractions those days and all that, purifications of RNA. So he said, now you have to give a seminar. So he called everybody. There was one Professor Satcher was called from IRI. He was a great biochemist. So they were all sitting like all of you, and I was to talk. So I was very interested. I said, "Bahut padh ke aaya hu main." So I started and I said, you have to use TCA. Normally for precipitation you use TCA. And I said, you use TCA, you precipitate, you take the RNA out, you will run it and all that. So he stops. Why do we use TCA? I said, this is what they have used in TCA. He said, I don't have TCA in my lab. I have hydrochloric acid. Can I use it? That's also an acid, this also an acid. I was blank. I don't know, many of you may not know the answer, but I don't know. So this is the kind of teaching which is there. So he said, your lecture is stopped. You can't move forward from this point onwards. Go back to library, read, come back. Then after 1 week, we'll restart the lecture. Everybody was asked to go. मैं घर गया, I was living with my brother in Lodi Road. मैंने कहा मैं तो PhD छोड़ दूँगा, सबके सामने तो ऐसी बेइज्जती हो गई कि कुछ नहीं है. So this is how they used to teach. The second thing, in fact, his father, Professor Maheshwari, was the main Professor Maheshwari. He used to give a lecture. The title was How to Give a Bad Lecture. So that was how they used to teach. Many teachers, if you find, then those days we never use. I still don't like to use a PowerPoint presentation in the classroom teachings. Whiteboard, blackboard is something. Now many teachers, what they do, they have a chalk in one hand and duster in another hand. So they will write something here, So by the time the student has even looked at it, वो rub कर देते थे. So this is, you learn yourself. You said, what you have to write. I mean, when you prepare your lecture, you must have in your mind what is to be written where. You have to design your blackboard, whiteboard. What you can rub, after 10 minutes, after half an hour, or something which need not be rubbed at all till the end of the lecture. Because that is the main message which you want to convey, and that message should stay in their eyes till the end of the lecture before they go.
So design.
So design, the teaching is a art. So we learned by that self. In fact, I was so happy when I was the Vice Chancellor. I was inaugurating something for foreign, this is Northeast students. And they had invited who was IGP in Northeast, placed in Delhi. So I didn't know, I didn't remember him, So when he came there, he said, "Sir, do you remember me?" So I said, "I'm sorry, yaad nahi hai bahut." I think this was in 74, 75. And I had taken your class. He was a life science student. He said, "I've taken your class." After that, I went to IPS and all that. I still remember your auxin lecture, the hormone lecture which you taught. Shall I tell you what I learned? I said, "You remember after 30 years?" He said, "I can't forget it." No, that was a great compliment to me. That people remember. He said, "वो मुझे अभी blackboard याद है." That's what I'm talking about, blackboard. He said, "वो blackboard याद है, वो I can still remember." So teaching has to be done in a very different way. It's not passing on the information which is present in the different internet पे information है, books में है, or it is even in publications. A teacher is not a transmitter. That you transmit what is published. वो तो मैं खुद भी पढ़ लूँगा.
Right.
In fact, I used to bunk a class in my masters because वो dictate करते थे teacher.
Right.
He would have his notes prepared. बता दिया, हर साल बता दिया. मैं तो करता नहीं था class attend. Because I thought I was not learning anything. It's better I spend that 1 hour in library and read something more. So teaching I still enjoy because I learn. Not because I'm teaching, but I'm learning in the process. और students को मैं कहते तुम पास तो हो जाओगे. If you are coming here just to clear your grade, that is not what you should be looking at. And I said I will tell you the question paper. अब कैसे बताता है question paper? I said each one of you will ask for each lecture, ask 5 questions which nobody else has asked about the science. तो 20 लड़के बनाते थे 5-5 क्वेश्चंस, 100 क्वेश्चन हो जाते थे. ऐसे इसी 100 क्वेश्चन में से 5 आएंगे. सो एटलीस्ट 100 क्वेश्चन तो पढ़ते थे. तो वो सब उनके दिमाग में होता था. उसमें से ही 5 आने तो क्वेश्चन पेपर में पहले बताता था. बट दे सेड इट वाज वेरी टफ कि वो 100 तो पढ़ने पड़ेंगे. Different methods. It's an experiment by itself. Teaching is an experimentation. As I was telling you, when I came to ICGB, then the whole group was working in a different area on chloroplast biology, but I thought we had to do something which has a translational effect because it's biotechnology institute, so something product orientation has to be part of it, basic work linked to product development. So the whole group was reorganized into stress biology program, and all of them cooperated to leave their areas and come into this, although everything was networked together. So we thought that Keller's cells is also a stress cell, and whether if glyoxalase is involved in there, so whether glyoxalase would be also involved in the stresses, and that's why the whole story of this worked. By the time we had a student and we were able to produce antibodies to this one, we started screening the libraries, we were able to clone the genes, and finally showed that when you give plant stress, whatever stress, it could be salinity stress or it could be drought stress, that means less water or more sodium chloride. And under those conditions, the expression of glyoxalase 1 and also glyoxalase 2 goes up. That was found later on glyoxalase 2. So there was, earlier we were looking at light, nitrate reductase and nitrate, 3 things which were working in order to induce gene expression. Now here we are looking at stress and glyoxalase 1 and 2. So the whole thing was there. Perception mechanisms changed, although light is always there, but perception mechanism here was inducer was stress. And looking into the expression of the genes. And that's what we started working on this. Now, how do you show that these genes are linked to stress? So, we made transgenic plants where we took Glyoxalase 1 gene, Glyoxalase 2 genes under 35S promoter, which are general promoters, and then you express more of these proteins. And when we express more of these proteins, we found that these plants were able to grow under stress environmental conditions. Then we asked many questions: how does it happen, the biochemistry behind it, the other changes which are occurring, and that is what has kept us on in this field for a very long time. But in short, what our whole model which worked out was, till that people had been showing that under stress, one thing happens is you get more of reactive oxygen species. You know, all of you know reactive oxygen species which are produce oxidative stress. There is also some hormones which are changing in plant systems. One of the hormone is called abscisic acid, which is a stress hormone. So, those were the kind of signals which people had been working on. So, what we found based on number of experiments which we had done, that in addition to these signals, when plants are under stress, they also increase methyl glyoxal. Okay? And methyl glyoxal, once its level increases beyond a certain level, then it can induce the expression of glyoxalase 1 and glyoxalase 2. Okay? And once that is induced, these two proteins will be produced and they will detoxify methyl glyoxal. So, it is a survival strategy for the plants. I think that is the model which I have. Maybe you can show it to them later on. And so, this we thought Mg is a signal molecule, a new signal molecule which is there. Of course, people are not talking about Mg as a signal molecule even in animal systems and yeast and others. So, this is more work is coming up on this. And we also did a total transcriptomic profile of different rice plants in different conditions respond to Mg, and we find it is not only 2 of these genes which are changing, but many other genes which are changing in response to methyl glyoxal. So, that is the great broad model on which we have been working, but this model became even more difficult. So, this model took me back to my earlier one. So, you know, light, reductase, nitrate. Nitrate was a substrate, substrate was inducing. So, 3 things which are working. I came back to the same thing, is a stress, glyoxalase, and the substrate of glyoxalase is methyl glyoxal, and methyl glyoxal also induces. So, we are coming back to substrate inducible genes regulated in the one case by light, here in other case by stress. So, the model which we worked long back turned out to be a similar kind of model, because there are not too many substrate inducible enzymes in the plant systems. So, that is why these models become very interesting, how these 3 different signals
coming together
work together in order to induce the expression of the genes. So, we are not later on not only in glyoxalase, but many of the people usually have a tendency to look at genes which are upregulated. Up is always, everybody wants to go up, you know, in life. So, want to always work on those which are upregulated genes. So, I said we should look for downregulated genes, and this was the similar model which I had for phytochrome, because phytochrome gene is downregulated. So, I also wanted to look for some genes which are downregulated, and one of the student Pradipta, who worked on RBS32 gene, and that was downregulated under stress under the similar conditions. So, we created a similar model as which we have earlier, and that downregulation we found was also due to histone changes, the chromatin modification changes that we published that work earlier. So, how these gene expression changes are going on, all of you know, but I am trying to tell you the kind of models which you have which need to be worked out in more details to see how these different signals which come from different places interact, crosstalk and give a response. That is what basically means. But the story became little complicated of glyoxalase 1 and glyoxalase 2, because in rice, when the whole genome sequencing was done, you do not have only one glyoxalase 2, there are many genes involved in it. And so, the students started working on all of these. It turned out that not all of them are glyoxalases. They do not have activities, but there are 3 at least which have good activity. And one interesting observation which was made, because most of these glyoxalases are metal dependent enzymes or metal stimulated, and they are stimulated by zinc, most of them. But what we found was one of them which was stimulated by nickel, and that was published that you also have a nickel dependent glyoxalase in plant systems. So this story is still continuing. There are some which we have now found which are present in the nucleus. There are some glyoxalases which are specific only when panicle development takes place, and so on and so forth. So one of the major area of activity along with Dr. Sneh, because when we first published our paper in Plant Journal, that was the first paper in country from Plant Journal with a cover page and all that in 99, and then Sneh joined the laboratory, and then I asked her whether she would be interested. She continued, she went into the glyoxalase 2 story, and that PNS paper which we published was where both of these genes work together. And since then she has been, she does many other things, but also works on glyoxalase pathway, work together on this.